we'll talk about a very important subject in physics and clinical application of an MRI imaging. Today we'll talk about diffusion imaging and ABC. Let's start by definitions. Okay? Diffusion weighted images is concerned with the diffusion phenomenon, which is the well-known physical phenomenon that describes the free and spontaneous and random movement of the micromolecules within a medium, special media or specific media, under the effect of heat. Okay? This free movement of the particles can be subdivided into three categories. It is called, by the way, Brownian motion, related to someone who noticed this motion, type of motion, and it's called after him. The first slide describes the type of movement of the micro molecules or particles in a clear water medium or clear fluid medium. You notice that this type of movement is totally free movement, totally random, totally spontaneous. No limitation in any direction for the movement of the particles could be seen in this type of movement, and it is called the isotropic diffusion. Okay, this is the first type of movement without resistance of any type. The second type of movement is called anisotropic diffusion, which means there is also free movement of particles within the medium, but
preferred
in
direction,
certain limited and allowed direction. The direction most of the time is longitudinal direction, since we are talking about the brain, and the boundary or the limitation for the movement is due to lines or sheets or tracks, and the most common example for this type of barrier is the brain white matter tracts, corticospinal tracts. As you noticed in the brain, it will pass longitudinally from the cortex down to the spinal cord, and it forms barrier to the movement, which is free for the particles due to the myelin sheath and fascia and fibers itself. But this type of movement is called anisotropic diffusion, which means there is limitation in one direction of the movement. One direction is allowed and other directions are limited due to this barrier. The third type of diffusion is called the restricted diffusion. You see in this slide a lot of microorganisms in the tissue that limits the free movement of particles within the medium. This type of organisms could be cellular, could be connective tissue, could be other particles, but they are solid particles that markedly limit the diffusion ability of the molecules or particles within that medium. Example for this type of restricted diffusion could happen in a lot of pathologies. One of the most common pathology is stroke, acute ischemia of the brain. What happens in acute ischemia? There is deprivation of oxygen, there is limitation of permeability, there is intracellular accumulation of a fluid,
yes,
and shift of a fluid from extra to intra with diminished extracellular fluid. So there is a real barrier to the movement in all directions due to these multiple factors. Another example of this type of restricted diffusion in the brain is due to tumors. Tumors are in different pathology. We have a high cellular amount and concentration in the tissue, which makes it very limited to the free particles to move freely inside the tissue due to high cellularity. The first one is stroke due to permeability and swelling of the cells. The second is due to high cellularity. The third example in the brain is due to inflammatory, and we are specifically talking about abscess. Abscess in the brain will make a media which is highly viscous due to inflammatory exudation and production and secretions, which also prevents the movement of particles freely in the media. So these are three mechanisms of restricted diffusion in the brain. Knowing these facts, if we can measure the diffusion capability and whether it is free or limited, which means restricted, by a way of imaging and by a way of sequential MRI application, we can depict the pathology specifically early and helps in diagnosis and treatment subsequently. Okay? The aim of the diffusion and the ADC mapping is to depict and to analyze this type of movement, whether it is totally free, not restricted, or restricted, to show it as a grayscale, bright or dark, and subsequently helps in and aids in diagnosis. Okay, this is the basic cause of this type of sequences, and what in thinking we will do to generate this type of sequence. Now we will talk about the practical and technical and physics of this issue or matter. The second part of the lecture will talk about the clinical application of this sequence. I'll divide the lecture into two parts, clinical and physical. Okay? Let's start by putting these three types of diffusion. Remind you, the isotropic, the anisotropic, and the restricted diffusion. Also, before going to the grayscale, we have to say one information which is important. We can depend on one trial for detecting the diffusion capability. It is some sort of accumulative process. So we have to repeat the sequence multiple times to achieve the true results. This gives you an example. If you put a drop of ink in water, if you catch the diffusion in the early minutes, it will not be true. It's just the minimal distribution of ink within the liquid. If you delay it little, it will be more accurate. If you delay it more, the particles will diffuse, fill the cup, and give you the result that you will say, yes, this is an ink or a cup filled with ink. Okay, it is diffusely filled with the substance. Okay, that basically we have to depend on multiple sequences, not just a sequence. You will notice what I mean by this multiple sequences when we talk about the physics. The second point, we have to agree that in the final phase we are talking about bright or darkness. We will give the brightness to determine the high diffusion capability and the free capability of diffusion, and the darkness is to determine the limitation and restriction of diffusion. Let's go in